So we're going to go into the specific effects of insulin, and first on carbohydrates. So there are really a variety of things that get activated, a variety of things that get inhibited. What gets activated is glycogen synthesis and glycolysis, two processes that consume glucose because there's plentiful glucose in the bloodstream. Okay? The reverse processes, glycogenolysis, which produces glucose, and gluconeogenesis, which also produces glucose from smaller molecules, are inhibited. And that's specifically important in the liver, because the liver is the only tissue that has gluconeogenesis. In both liver and muscle, you have a decrease in glycogenolysis, degradation of glycogen. The tissue-specific effect of insulin is this last one. This should be really highlighted, very important. Insulin stimulates glucose uptake in these specific tissues, muscle and adipose tissue, by increasing the number of GLUT4 transporters on the cell surface, in the cell membrane. So more transporters, more glucose taken up into the cell. Adipose tissue, or effect of insulin on lipid metabolism. Let's start with adipose tissue. You remember, adipose tissue is where we store a lot of tags. The adipocyte is roughly 80% tags. Okay? So adipose tissue, important effects on adipose tissue is first to decrease lipolysis by inhibiting hormone-sensitive lipase. Second is you increase tag synthesis. And how are tags synthesized? That you need fatty acids and you need glycerophosphate. The fatty acids are coming from the dietary tags from chylomicrons. Lipoprotein lipase breaks them down, picks those fatty acids up into adipose tissue. Then they are esterified to glycerophosphate. Where is the glycerophosphate coming from? It's coming from DHAP from glycolysis. So remember, adipose tissue picks up glucose. It's using glucose for its own energy needs. So it will have an ample supply of DHAP and glycerophosphate as a result. And that's the backbone used to esterify fatty acids from the diet, or from de novo synthesis from VLDL, and you increase tag synthesis. One important positive effect of insulin, this is very important, is that it can upregulate lipoprotein lipase. This is a distinct lipase from hormone-sensitive lipase. It's distinct in its location and it's how it's regulated. It is part of the endothelium, so it kind of hangs out attached to the endothelium wall, if you want. And the enzyme can access the lumen of the bloodstream and access chylomicrons and VLDL. So it's choose on those to be able to pick up fatty acids to be stored in the respective tissues. used or stored. Okay, so this is important effect of insulin. In most tissues, as I mentioned, so here we have TAG increase, synthesis of TAG, decrease lipolysis. The other class of molecules, proteins, you're going to basically increase protein synthesis in most tissues because there's plenty of amino acids in the bloodstream when after a meal, and all the tissues will try to pick them up to replace what was turned over in a prior cycle. Importantly, though, the muscle will use up a lot of those amino acids because of its mass. Okay? But as I mentioned again, you have three things that are stimulated: glycogen synthesis, TAG synthesis, and protein synthesis, all by insulin. And this is really a summary table of the effects of insulin. Very important. Without getting into all the details, but here's the first important effect. Note where the checks are. Muscle adipose tissue only, not the liver. The liver will pick up glucose, but not because of insulin, because of high glucose. The liver transporter is GLUT2. And that is a low affinity transporter. That means it doesn't have a high affinity for glucose. So, but when glucose rises in the bloodstream, the liver will start to pick up more of that glucose. Okay? Cellular respiration, what does that mean? If you don't understand what cellular respiration, maybe you missed a couple of lectures. That's the ETC, electron transport chain. That's where we're using oxygen. Why do we need the ETC? Because most of the metabolic pathways, catabolic pathways that produce energy, like glycolysis, PDH and TCA, produce NADH and FADH2. And you need to take those high energy molecules, oxidize them through electron transport chain to produce ATP. So as a general rule, insulin is going to stimulate cellular respiration, production of those high energy molecules and their eventual production of ATP. Stimulating glycogenesis. That's in liver and muscle. Inhibiting glycogenolysis. That's in liver and muscle. Stimulation of amino acid uptake. The muscle is a key tissue, but really there's going to be increased uptake in other tissues as well. Protein synthesis is stimulated throughout most tissues, inhibiting protein degradation. TAG synthesis is specifically increased in liver and adipose tissue. You inhibit TAG lipolysis, specifically by inhibiting hormone-sensitive lipase in adipose tissue. And this is really important in that this allows lipoprotein or lipase is responsible for accessing fatty acids from circulating tags in the form of chylomicrons and VLDL. There's an upregulation, meaning there's an increase in the amount of lipoprotein lipase on the surface of endothelial cells. Okay, so glucagon is going to have, again, its own set of effects on metabolism. The effects on carbohydrates primarily are to increase glycogen breakdown, glycogenolysis, by activating pKa phosphorylating glycogen phosphorylase kinase, phosphorylating glycogen phosphorylase, and that results in glycogenolysis. Okay? Simultaneously, it will inhibit glycogen synthesis. That's always very important. You're activating one thing, inhibiting another. So you're not having both occur simultaneously. And the inhibition of glycogen synthesis is because you phosphorylate glycogen synthase by pKa, which becomes inactive under those conditions. You have an increase in gluconeogenesis. Okay? Both of these are key functions, again, of the liver. This is really a key thing here. Let me see. I'm going to change the pointer. Makes it easier to highlight. Not really. Okay, yeah. All right, so see this here, really important. These are the key functions of the liver. So think about it. Glucagon is high when glucose in the bloodstream is low. That means you are in a fasting cycle. So the liver's function is going to be completely changed, meaning it has a new function. Its new function is going to be producing glucose. Okay? How do you produce glucose? Through either glycogenolysis or gluconeogenesis. What happens with lipids? You're going to use whatever tags that were stored before, either in adipose tissue or the liver, as a source of fatty acids. So you break down tags. Lipolysis will be increased primarily by activating hormone-sensitive lipase. Okay? But I want to kind of point something out. Adipose tissue doesn't have a glucagon receptor. So hormone-sensitive lipase is activated in adipose tissue by epinephrine through the beta-adrenergic receptor. Okay? So it's a unique and important kind of detail to pay attention to. So you have fatty acids are going to be used as a source of energy instead of glucose in the liver, in other tissues, in the muscle, in the heart, in many tissues that can use fatty acids, but not in the brain and not in red blood cells. Okay? These two organ and cells still need glucose as their major source of energy. Now, what's going to happen as well is that the liver will be increasing ketone body synthesis, ketogenesis, because it's using fatty acids as a source of energy, producing a lot of acetyl-CoA, and some of that acetyl-CoA is used in ketogenesis. So note how important the liver is. It's providing glucose, providing ketone bodies, two different forms of energy to other tissues that can use those forms. In terms of gluconeogenesis, what's going to happen, where the building blocks or the precursor molecules for glucose coming from? They're going to come from muscle amino acids. So in the liver, there will be increased uptake of amino acids. Again, muscle protein turnover. It doesn't mean that you're wasting away. It just means normal muscle protein turnover. Breakdown of muscle proteins gives you amino acids. Amino acids released in the bloodstream, picked up by the liver. So this is amino acids coming primarily from turnover of muscle proteins. Okay? And they are going to provide the carbon skeletons for gluconeogenesis in the form, we talked about one of them, alanine can be released, goes to the liver, becomes pyruvate. Pyruvate can be used to make glucose. So here's a summary of the effects of glucagon. Pretty straightforward. Without going through all of them, here's mainly effects on the liver. Activation glycogenolysis, inhibition of glycogenesis, activation of gluconeogenesis. This one here, note, no effects on muscle. Really, no effects on adipose tissue. This is an effect of epinephrine on the beta adrenergic receptor that in turn activates adenylate cyclase in adipose tissue, producing cyclic A, activating pKa. pKa phosphorylates hormone-sensitive lipase that breaks down the tags in adipose tissue and make those fatty acids available to the rest of the body. Okay, so that's why it has a different color. This is due to epinephrine. Okay. Contrast the effects of glucagon to the effects of insulin. So insulin can affect basically all three tissues in distinct ways, whereas glucagon effects are pretty much localized to the liver. Okay, so which gets me to a point here, an important condition, but I want to just remind you that the reason glucagon is released is that, as we discussed yesterday, the brain senses a drop in blood glucose. So it sends signals to the pancreas to increase glucagon release. It sends other signals to increase epinephrine synthesis, cortisol, and so on. So the key there was a drop in serum glucose. So that's the normal body response, is sense or notice a drop in serum glucose, you respond by producing glucagon, epinephrine, and cortisol. If the body doesn't respond appropriately, you're going to have a condition called hypoglycemia, meaning a decrease in normal serum glucose. Probably anything below, say, 70 milligrams. The normal concentration is somewhere between 80 to 100 milligram per deciliter. So if glucose drops significantly, say to about half, you're going to its normal concentration, 40 to 50 milligrams per deciliter, you're going to, the body will start, that again has to do with that brain sensing the drop. And usually the body can respond in one of two ways. So the first symptoms are called adrenergic symptoms. So you get anxiety, tremor, palpitations. That's the brain telling you there's not enough glucose in the bloodstream. And usually the release of glucagon and so on ensues. But if it doesn't, it becomes even worse. So you're going to get confusion, aberrant behavior, could even lead to coma. So there's really two ways that two types of symptoms can result. For the first one is the more common one, that the body is trying to respond by the brain is trying to respond by increasing, say, glucagon and epinephrine. In the second case, these symptoms or the drop in glucose happens so gradually that the brain doesn't even register. So they can be much more serious, and they can result in seizures, coma, and that's because usually of a slow drop in glucose that kind of the brain does not detect. But anyway, as a general rule, hypoglycemia can be easily treated with glucose, either oral or IV glucose. Within a few minutes, everything gets back to normal.